Good evening, Pittsburgh and universe. This is JC on a bike. My name is JC and this is Journal Club on a bike. Tonight we're going to talk about a paper by the Anne Churchland lab, but we're going to mention a few other papers, especially a few by Ken Harris's lab, and try to address a pretty big trend in neuroscience today that, from the perspective of many other fields, is a bit shady. And Anne Churchland's paper is sort of an example of, potentially anyway, what could be a very cool way to take advantage of the tools that we've been building in terms of interpreting big data and giant data sets of recordings from hundreds or thousands of neurons. But the basic gist of all these kinds of papers is that the animal will perform a task, and then either with imaging or large multi-electrode probes, you will record neuronal data and then try to correlate the neuronal data with something that you can observe in the behavior. With place cells, you're correlating the location of the animal to the place where the cell fires, and that correlation is very high. Look at this person. That correlation is very high for place cells in familiar environments, and it's lower for environments where the animal has never been. And so those correlations are often believed. Place cells and grid cells indeed correlate with locations in space, and the correlations are fairly strong. And manipulations of the environment or the experience in place cells and grid cells excuse me. But the correlation measured on grid cells and place cells, it's a very high correlation. Sorry about that, thank you. And with these behavioral studies, when they record from thousands of neurons and then they correlate neuronal behavior to some aspect of the behavior, these correlations are much lower in general. And they can even be orders of magnitude lower than the correlations typically considered significant for place cells and grid cells. But nonetheless, there are tens or even hundreds of papers nowadays that are using imaging or other means of recording many neurons at the same time, and then correlating the activity in those neurons to particular landmarks in the experience when the animal was doing this or doing that, to try and discern how signaling in the brain works. Now, the problem with these experiments, and this is the issue, that we have these beautiful techniques for recording from hundreds or thousands of neurons, but we still don't really have a working theory of how the brain works. So, putting these signals from thousands of neurons into the context of the animal's experience is difficult, because the only readout on the animal's experience is that which we can observe. And so if you can't observe it, what state is the animal in? What is the animal experiencing now? Stress, excitement, happiness, arousal? we have no way of knowing whether the animal is cognitively processing the task we put in front of him. We just know that we have a behavioral readout. But without a good idea of how the brain works, or why a neuron would fire action potentials into relation to something like, in this case, the holding of the handles, it's not very clear what their theory is based on. And they don't care because they don't do any manipulations to test the assumptions that they make. And so the way this paper works is very simple. They have cleared the entire skull so that they can image the whole upper layer of the cortex. So they're basically imaging apical dendrites and cells in superficial layers. And from this calcium signal, they get a kind of idea of what the microcircuit is up to, at least when it's active. And the animal is trained to put its paws on two handles to initiate the task, and then some undetermined time after the initiation of the task by the animal pushing on the handles, a visual stimulus or an auditory stimulus, depending on how they're being trained, is presented on either side of the animal, on the left or the right. And the stimulus is presented twice. And then the animal is required to lick the appropriate water spout for a water reward to indicate that it knows which side the stimulus was presented on. And it's interesting because the task itself is quite trivial. They don't really care about it. It's just a means to an end. This task has certain instructed, quote-unquote, activities that they're supposed to do. They are instructed to hold the handle. They are instructed to lick. So, these activities, the instructed activities, are separated from the uninstructed activities, like grooming, or whisking, or scratching with their hind paw. And so in figure 1, they show you the basic setup of in the first two figures, they show you the basic setup of the experiment or the activity that the animal is going to participate in. And they show you a cartoon which describes instructed versus uninstructed, but keep in mind that uninstructed can be anything. And in future figures, we're going to take uninstructed movements from the video itself, and we'll end up using pixels that are mostly just the edge of the face. But anyway, I digress. So the task is simple, and then what they're going to do is just image the cortical activity in and around brain areas of interest. If you could get off your phone in time for us to move. Brain areas of interest to see what's going on in those brain regions during this task. And what they will find is that, for visual stimuli and auditory stimuli and the touching of the handles, and even when the animal sniffs, there are events which correlate with the cortical activity in kind of an expected way. When the visual stimulus is presented, V1 is activated. When the auditory stimulus is presented, the auditory cortex is activated. So some of these things they follow, right? And the correlation isn't surprising. But remember, how much of this activity can be described by or explained by these events is actually quite small. The vast majority of the activity in the cortex is not related to the touching of the handles or the licking of the completing of the task or whether it was successful or not. But what we're going to see in the first figure is just how they do it. And then in figure 2, what they're going to try and show you is how they're going to extract the event kernel for particular uninstructed movements and instructed movements. You might think that, okay, we're just going to do a linear model. Why is this person not turning? I don't know. Shoot. But they don't do just a linear model, even though a linear model is dumb. They do a linear model plus this event kernel, which is just a sliding window of probability which tries to describe how much of the cortical activity is readily explained by a particular event kernel, in this case whisking in figure 2. And so they show you how whisking can be converted to a binary, or sort of a binary thing, and then using that as a time of the event, they can then make event kernels and look at how thank you, sir. Event kernels, and they can look at how the activity relates to these event kernels across time. And for whisking, for example, they see in this example in figure 2, they show you that when the whisking occurs, there's a signal in retrosplenal cortex that precedes the barrel cortex. And the other thing they demonstrate here is that a linear model of the activity isn't sufficient to describe the fluorescence change. Analog variables are not enough. Which again, it's not really surprising, but at least they're finally talking about it. So figure 3 is going to look at various specific kernels and show you how powerful the kernel is relative to just an analog variable. And here they're going to look at particular kernels, handle hold, V1 stim presentation, nose sniff, and show you that if you do the event kernel around the sniff or the event kernel around the handlebar grab, that you get expected activity in expected places. But even more surprising in figure 3 is that they show that variables taken from the video variables derived from the video, up to 200 variables can be derived from the video itself, and using only 6 of those variables you can already capture 90% of the variation in the calcium signal that they see across the cortex, with only 6. And those are drawn from the picture that you see with the highlighted areas in the video. Those are mostly hairs and mostly outlines of the face to show you whisking and face movement. That's a spectacular result, because all of the variables that they're interested in, like the handle grab or the licking, are able to only describe a very small fraction of that calcium signal, a very small fraction. The rest of it simply is not explainable by task variables. And instead, again, 6 variables from the video is enough to capture almost 90% of the activity. And that's spectacular. But what does this really mean then? Well, it means that there's probably been a lot of papers where we've talked about correlation, but it's not meant anything. And we'll see that more as we get deeper into this. Because the next few figures are just going to hammer this point home, that the uninstructed movements, the stuff that the animal does besides the task, is way more useful in understanding the activity in the brain than the task variables. So figure 4 is going to do an interesting thing. What it's going to do is going to take the whole variable, the whole model, which explains the data very well. And then what it's going to do, come on dude, green. He's going slow on purpose, you can tell. So figure 4 is going to take individual variables and look at their contribution to the whole model by shuffling them or removing them from the whole model to see how much of an effect the single model has on the whole successful model. And then they're also going to look at how this model, this variable by itself, contributes to explaining the data. And what they show here is that shuffling any of the task-oriented variables does 0. One particularly cool one that does almost nothing is time. Which I think is quite surprising, because you would think that time, because this is a task, time would be something that would figure in. But it doesn't. And in fact, when you shuffle the time variable or remove it, it doesn't significantly change the effectiveness of the model. So figure 4 is cool because it really breaks down how weak the correlation is for most of the variables that historically this paper would have been interested in. In Ken Harris's lab, they do experiments often in the visual cortex, often just with Gabor gratings. And in that sense, I think you could argue even more that it's probably just correlation fishing without any real testable hypothesis behind it. And I think that's what this paper in the end is trying not to be. So figure 5 is going to look at how some of the variables are, and some of the non-instructed variables tend to be task aligned and contribute to that time during the experiment, those signals. So it's similar to how they were trying to do the unique contribution in figure 4. In figure 5, they're going to try and sort out what contributions are made in and around the task versus not related to the task. And what they show is that for single trials, for the average, the task variables do okay, you know. But for single trials, the model's awful unless you use uninstructed movements. And those individual traces there those are showing that the only model that's able to capture the network dynamics in single trials is the model that uses the uninstructed behavior. The task-aligned variables just can't do shit for the single trial signals. So figure 6 takes the data from figure 5 and looks at how the data changes as the animal learns. And this figure seems to indicate that even the uninstructed behaviors become more stereotyped. And so can actually start to contribute to the correlation that you see as a result of the animal getting better but still not related to the actual task. And this gets back to Adam Hatman's papers where the animals are doing funny repetitive behaviors to learn a delay time between lever presses. And then you're left wondering from this paper, are we really looking at neurons that are involved or just animal neurons that are involved in this repetitive movement thing? And it turns out the predictive power of the uninstructed movements goes down. Fuck you. See it? Fuck you. Figure 5 takes this data to the 2-photon imaging level so that we can image individual cells. And this I find the interesting figure, because this figure could be used as a way of sorting out cells that seem to be more relevant for the things you're interested in. But, and there's still a huge but here, that means you're still working on the assumption that action potentials mean something like holding the bar or licking the spout or other things, that other aspects of the experience that we can identify. And I still think that's a very crappy assumption, because it rests on the idea that that's how the brain works, that neurons fire action potentials as bits to indicate what the animal's doing. And there's no reason, we have no evidence to believe that spikes can tell us those things. We have no evidence to believe that holding the handle should be encoded by single neurons. We have no evidence to believe that any of the things that they're correlating with neuronal activity should actually correlate with neuronal activity, much less linearly, which is another assumption in this paper. If you have a linear model of action potential activity and neuronal activity, you think that neuronal activity scales linearly to things. And I don't think we have very many reasons to believe that. And so figure 8 takes data from a neural pixel probe and, using, I would presume, similar analyses, states that they find similar correlations between movement and neuronal activity as opposed to something else task related.